everyone. This is my thesis defense in preparation. My topic is titled Nanotechnology's Role in Developing the quote-unquote Google Maps of the Human Body. In other words, the Human Cell Atlas. My name is Sohum Punj. I am a junior at Colleyville Heritage High School, and I live near the Dallas area. So just a bit of an introduction about this topic. An overview about the Human Cell Atlas. So the Human Cell Atlas is basically a global initiative to create a comprehensive map of all human cell types. It aims to revolutionize our understanding of cell biology, and it's got contributions from over 3,600 researchers across 102 countries. So this topic is very new, which means that not much research has actually been done regarding it in the science world, which I will talk about later. So my topic was basically how can I input nanotechnology into the HCA, the Human Cell Atlas. And nanotechnology can enable high-resolution imaging, ultra-sensitive detection, and precise sorting of cells using advanced tools like nanoscale imaging, nanobiosensors, and nanofluidic systems to facilitate cellular mapping and analysis. A little bit of the significance about this research: it provides insight into the cellular heterogeneity into the human body and supports the development of precision medicine and therapeutic strategies. So this is my literature review. So a little bit of a historical background. This project, of course, started with the discovery of cells by Robert Hooke in 1665, then turned into the development of the cell theory. And now it's more about advances in single-cell analysis and genomics. So if we dive into cell biology for a minute, the three categories that I kind of used to talk about were cell mapping for nanoscale imaging, nanobiosensors for cell analysis, and nanofluidic systems for cell sorting. So going into cell analysis just for a minute, my literature review focused on one of these articles titled Nanotechnology Enabled Biosensors, a review of fundamental design principles, materials, and applications. It taught me a lot about biosensors and even gave me some background information to cell biology as a whole. So about my methodology, my research approach was secondary research utilizing peer-reviewed journals, academic books, and digital media. I used critical analysis of nanotechnology applications in cell mapping, analysis, and sorting. The three techniques that I mentioned earlier that I studied are nanoscale imaging, in which I examined the expansion microscopy, fluorescent quantum dots, and atomic force microscopy. In biosensors, I focused on carbon nanotubes, which I actually picked this topic from one of Professor Lilova's lectures, so I found that one really interesting. Graphene-based nanosensors and gold nanoparticles. And the third I decided was nanofluidic systems, in which I studied sorting through physical characteristics, dielectrophoretic sorting, and hydrodynamic sorting. So, like I said, a lot of this research hasn't been done before, so what I did was I focused on these three topics, and then in the things that I mentioned just now, I went a little more in depth into each one and studied a little bit more about those. So my key findings I found with regards to nanoscale imaging: it improves visualization of cellular structures beyond the diffraction limit. For biosensors, it enables real-time, highly sensitive molecular detection. And for nanofluidic systems, it provides high throughput and precision cell sorting techniques. Just a little bit about the comparative analysis. So I identified the advantages and the limitations of each of these technologies, and then I evaluated the potential impact of these nanotechnologies on the Human Cell Atlas development. With regards to their implications for medicine research, the Human Cell Atlas enhances disease diagnosis and can lead to more precision medicine. It provides tools for better understanding cancer, immune response, and regenerative medicine. And in terms of comparative analysis, expansion microscopy is effective for high-resolution imaging but requires complex processing, whereas quantum dots provide a long-term tracking but have potential psychotoxicity concerns. And then about the others, graphene-based nanosensors offer ultra-sensitive detection but require improved standardization for medical applications. And nanofluidics incorporate high efficiency in sorting, but it requires further miniaturization for scalability. So just to summarize my conclusions a little bit, nanotechnology is currently revolutionizing the construction of the human cell atlas, especially with the incorporation of the techniques that I studied. Advanced imaging, biosensing, and cell sorting techniques enhance cellular analysis. And for limitations, I had to depend mostly on secondary data as, like I said, a lot of these articles didn't really have experience in the human cell atlas and really more just focused on the cell mapping or analysis or sorting aspects. I had a limited experimental validation due to restricted access to data sets, of course, obviously looking at free journals and articles. In terms of future directions, the integration of AI with nanotechnology for automated analysis is greatly beneficial for the future. Development of more biocompatible and cost-effective materials, and expansion of nanofluidic platforms for broader clinical applications. Some things about the gaps in my research. Like I said before, I had a limited access to experimental data sets, and the need for real-time imaging and improved accuracy in cell sorting was also quite prevalent. Lastly, I just want to say thank you to the professors that I worked with, Professor Lilova and Professor Virgil, and a special thank you to the Gifted Gabber team and to Coach Joe for allowing me this opportunity. This is something that I never thought I would research about, especially with just the complexity of the things that all of us here have talked about. But the professors really did an extensive and thorough job of making sure that I understood what I was talking about and really helped me and guided me along this way. So with that, I will end my presentation and be open to questions.
Yeah. Hey, thank you. Please stop sharing so that I can get Professor Lilova to ask you a question.
Yes.
Very interesting topic and very complex. So if you are to do the mapping, if you are to choose right now how to do it, what would you choose? Which technique you start with?
I would probably do expansion microscopy. I researched a lot more about expansion microscopy, and there was an article by Fei Chen that really talked about expansion microscopy in detail. So that was one that I understood to be the most efficient and effective in this category, especially because when paired with the other techniques used for nanoscale imaging, I feel like it would really improve the quality of cell mapping.
Great, thank you.
Thank you.
Excellent. A question from Professor Virgil.
Congratulations, Sohum. All right. So this is just a question that you can easily answer. So what did you study that made you want to conduct this type of, research project?
Yeah, so I'm currently in my high school biology class. We had finished up our unit on different types of cells in the body, and I was really inspired by the variety of cells and just the complexity of each cellular function and structure. And that really led me to look more into the different types of cells. And I came across this project done by a few professors, and it really intrigued me into looking at, well, what are all the ways that by identifying the functions and the structures of all these cells, we can further develop medicine and research in this aspect.
That's great. Thank you.
Okay. Wonderful. Thank you for presenting, Sohum.